Hi, my name is Cierra Sapora, and I collaborated on this research with Stephen Allison and Brianna Finley, whom are working with the University of California, Irvine. The research is called Biogeochemical Consequences Under Microbial Evolution Under Drought. For my introduction, evolutionary adaptation under drought conditions are thought to affect microbiomes. By isolating DNA strains from the ambient versus drought plots of the grass and shrub litter, we are able to identify the genetic changes. This research provides further proof of how climate change is affecting the evolutionary aspects of microbial communities in California. For my methods, I use FTIR spectroscopy, and I quantify the DNA material of litter and soil samples. As you can see in my figure 1, it is the DNA concentrations versus the litter inoculations and the differences between those. I then further use the Zymobiomics DNA mini prep kit to extract the DNA from the samples through a series of tests. These samples consist of grass and shrub samples, all from different plots located from the Loma Ridge site, such as samples 20 LRX GFC T1, which is shown to have 22.38 nanograms per microliters, contained on a scale of 2.16 over 2.00 for the purity of the sample's DNA. And just for the scale of the 2.0 scale, it's just there to show that if it's over that, it might be a little contaminated because you want it to be in the 2.0 range. For my results, based on the inoculations of whether their microbial cages were open or closed, it is found that shrub litter kept inside of the closed microbial cages had a higher rate of DNA concentration found and bacterial count per gram of litter. Furthermore, shrub litter that had the inoculum of being ambient and post-fire were found to have substantial increase of DNA compared to the assisted migration of shrub litter in closed microbial cages. This was found by doing DNA extraction and finding that the DNA extractions and bacterial counts gram of litter for both grass and shrub litters. For my discussion, originally my hypothesis was that if the microbial cages are kept open and in the ambient setting at the Loma Ridge site, then the litter samples will have a higher percentage of DNA compared to having these cages closed with the post-fire inoculum. However, due to the data results, the shrub outstood the grass litter unexpectedly, and especially when kept in a closed microbial cage and kept in an ambient and post-fire setting. So I can consider that perhaps because these cages were kept closed, that the moisture was originally in the plant was able to keep bacteria alive more than having the cage open and exposed to the natural weather in Irvine, California. Furthermore, perhaps the shrub has more DNA than the grass because the shrub itself has broader leaves, which means it has more surface area to keep moisture in and withstand the natural sunny drought environment at Loma Ridge in Irvine, California. Additionally, the grass litter is not evolving as fast as the shrub and thus is more susceptible to drought and succumbing to the natural environment. So in conclusion, bacteria and shrub plants that are not exposed to the natural environment are more susceptible to live. You can scan the QR code provided to see more information if you would like. Thank you.